Hello, people. Looks like a skeleton crew today. Only the good people came. That's all right. We're used to it. So I just moments ago uploaded today's notes and today's PowerPoint. So I'm sorry it wasn't there. I forgot to put it up yesterday. Spent, I would say, most of my life since we last talked working on this talk. Oh my God, put so much work into this one. So that's why I haven't graded your exams yet. I will I will have time today and tomorrow to grade your exam, so they should be done by Thursday. Um, and if you're in my illicit drugs class, Dr. Ramos, that's coming after. I'll grade that one after cardiovascular pharm, so that'll take even longer to get done. But it's been a crazy week here in sunny Pennsylvania. Always sunny in Pennsylvania. Is that what they say? I don't know. Okay. I'm not sure how long this lecture is going to be. I know I put a lot of time into it, but content-wise, it's not that crazy. I just wanted to make sure I was doing it right. In case you're wondering, one of the worst feelings in the world is to stand in front of people and talk about something you don't know anything about. That's embarrassing. Okay, so I put a lot of work into this one, and we will start in 3, 2, 1. Not really that fast. I'm working on it. So you can see how hard I was working on this. I got so busy, I never did a title screen, which I always like to do a cutesy little something there, but did not get to that. It's okay. It's okay. We don't need the cutesy stuff. But what we're talking about today is arterial smooth muscle. Arterial smooth muscle. I'm just pulling my notes up so I make sure I don't forget something. So at the beginning of your notes, I've got two different YouTube links of other people talking about smooth muscles. They're both good. They're both really good. They don't go into as much molecular detail as I do, but they're good, good reviews anyway. So when we talk about smooth muscle in this class, there's a lot of different places there are smooth muscles, but for us, what's most important are the cardiovascular system and the respiratory system. So mainly what we're going to be talking about, let me just give you a better picture to look at. You can look at that while I talk. So what are we talking about here? Nasal passages, you know, just the linings of your nasal passages. Those have got alpha-1 adrenergic receptors in them. So they're going to be innervated with the sympathetic nervous system. And sympathetic nervous system's almost always norepinephrine. There's one time when it's not, but so norepinephrine hitting alpha-1 adrenergic receptors, causing IP3 calcium system to act up, and that contracts those tissues, and they pull back away from the airway, and so you get, it opens up your airway. So those sprays you put up your nose, if you do that to open up your nasal passage, they're always alpha agonists. Okay, what else is important for the respiratory? We've got bronchioles. I don't know that much about the anatomy of the system. I guess it's like bronchioles come down to bronchi, or bronchi to bronchioles, or something like that. But anyway, they have got beta-2 adrenergic receptors. Beta-2, that's pretty important when we get into beta-1 and beta-2. So beta-2 adrenergic receptors are linked to cyclic AMP, and as we'll see today, cyclic AMP causes relaxation of smooth muscle. So calcium contraction, cyclic AMP relaxation. And those beta-2s then, when they're activated by your sympathetic nervous system or by epinephrine, will relax your airway, get your airway open so you can get more air in times of stress. Heart, in terms of the heart, well, I'm not talking about the muscle here, I'm just talking about the smooth muscle, the arteries that lead to the heart. Coronary arteries, they're all beta-1 receptors and cyclic AMP for relaxation. So basically, you never want those arteries leading to your heart to contract. If they do, you're not going to get blood to your heart and you're going to die. So yeah, we're talking beta-1 receptors, and that's key. Like, it's beta-1 in the heart, beta-2 in the lungs, because we've got different drugs for each. you don't want to relax one and contract the other or whatever. So we'll get into that eventually. Arteries. So arteries in your body go from like huge conductance vessels like the aorta down to arteries, which are still pretty big, and then down to arterioles, and then smaller and smaller arterioles. And the arterioles are really what's on a minute-to-minute basis controlling blood pressure and blood flow. So these are really important for cardiovascular pharmacology. And arteries all have alpha-1 adrenergic receptors. So because they're linked to the sympathetic nervous system and nothing else. There's no parasympathetic innervation of the arteries, only sympathetic, which makes sense because usually one contracts and the other relaxes. But here it's like one contracts, and if you want to relax, you just stop contracting because there's pressure in the vessel that will cause it to relax. So there's no parasympathetic innervation. But there are other ways to relax the muscle, and we'll get into that. The only difference here with arteries is arteries that lead to the skeletal muscle will have both alpha-1 and beta adrenergic receptors. And so this is really important. We'll get into this later in terms of norepinephrine versus epinephrine. Norepinephrine is more alpha, so your sympathetic nervous system will tend to contract your arterioles, but epinephrine is more beta, so it will tend to relax vessels going to your skeletal muscle. So if you're in a really stressful situation, you get a lot of norepinephrine being released, then it will relax the skeletal muscle, the arteries going to your skeletal muscles, which is really cool when you don't want to die. Okay. All right, so that's just kind of some background, and I will review all this again in this section because this section is going to be like this thing today about smooth muscle contraction probably should have been in the first section because it's more molecular. This second test is going to be more physiological. So like this section should have been in the first one, but I'm just like so far behind the 8 ball here. But anyway, all this stuff I just talked about with the receptors, where they are and what they do, that's going to be a lot of what this test is about. So this was just a brief introduction, but we'll talk about it a lot more. So anyway, with no more ado, I've had enough ado for today. Let's talk about the structure of the artery. So here's Mr. Artery, or Miss Artery, I hate to be sexist. The very inside, the hole, is called the lumen, and that's a pretty generic term used for whenever you have like a cavity inside a defined space. It's a lumen, so the blood is flowing through the lumen. The first thing the blood sees on the way out is the endothelium. So these endothelial cells are thin and flat. They are kind of protecting the smooth muscle from things that are inside the blood. I don't know how much we're going to get into coronary artery disease, but a lot of coronary artery disease is when you get damage to the endothelial cells, and that allows different growth factors to get into the smooth muscle layer, and then the smooth muscle grows into the lumen, and then that cuts off blood flow. That's part of the process anyway. So these endothelial cells are both protecting the smooth muscle from what's in the blood and also signaling the smooth muscle in terms of contraction relaxation. It gives off a contracting factor and it gives off a relaxing factor. So it's very important in controlling artery diameter. There we go. Okay. So subendothelium is just connective tissue, right? You always have to have connective tissue to keep things together. These are very thin and easily breakable cells, so this is holding that together. Then you've got elastic, and notice you've got elastic lamina on the inside and the outside of the smooth muscle layer. And that's really important too. We'll talk about how these arteries will take your blood from being, large swings in pressure down to constant flow eventually. So at the heart, your blood goes from, let me, hang on a second, I'm sorry. This is driving me nuts because I always talk with my hands. And with this background, you can't see my hands. Okay. Okay. How do we see? Okay, I guess I just turned it off. Okay, let's share again. Sorry, guys. Still learning ins and outs. There we are. Okay, so we'll talk eventually about how, like at the heart, your blood pressure goes from, if you're normal, goes from 120 down to 80, 120, 80, 120, 80, like this. As you get further away from the heart, that's going, the pulse pressure will decrease until you get to a constant flow. And that's being created by this elastic stuff. So when the blood pressure is high during systole, right, contraction of the heart, blood pressure is high, this elastic stuff will stretch, and then during diastole, it will recoil. And so that's kind of like, it's almost like a capacitor in electricity, where it's kind of like absorbing energy and releasing energy, absorbing and releasing. And that's going to decrease that pulse pressure until you've got a constant pressure when it gets to the tissue. Okay, that's your elastica. And then here's the hero of the day, smooth muscle cells, which are right here. So contraction and relaxation of these smooth muscle cells is going to determine the diameter of that vessel. And this is really vital in terms of controlling blood flow and blood pressure, because I believe it's the law of Laplace, although I've seen other names used for it recently. Excuse me. In the law of Laplace, it's kind of complicated mathematical formula, but in the end, it says that the flow, is it? The flow through the vessel is related to 1 over the radius to the 4th power. So any change in radius has a really big effect on the blood flow, because it's raised to the 4th power, which is, you know, a lot more than not raising it to the 4th power. Okay, so the diameter is really important, and that's being controlled by the smooth muscles and nothing else. A lot of inputs into that smooth muscle, and we'll talk about some of those today. And then you've got, you know, more connective tissue on the outside, tunica externa, or adventitia. So the adventitia here holding everything together. And important to realize that, you know, even though you've got blood flowing through this vessel, none of that oxygen gets to the smooth muscle from inside the blood vessel. The artery's got its own blood supply. So it comes from inside out, and then you've got these vasal vasorum, or vessel that will come out and feed those vessels. And also coming through this tunica externa is the nerves, the sympathetic nervous system coming through here and innervating these smooth muscle cells to control the diameter. Let's see, just checking my notes to make sure I did everything. Okay, so yeah, just backing up a bit to catch up on some of the things I may have missed when we went through. The endothelial cells release nitric oxide. We talked about nitric oxide. It diffuses out of those endothelial cells and into the smooth muscle cells, produces, what did I just say? It produces nitric oxide that diffuses out of the endothelial cells to the smooth muscle cells, which increases cyclic G, and that causes relaxation. So today we'll see the mechanism of relaxation there due to nitric oxide. It also releases endothelin, which is a protein, and it activates a G protein coupled receptor, turning on IP3 and calcium. So that's a contracting factor. So endothelial cells are going both ways. Okay. So your smooth muscle cells are kind of spindle shaped. If you look at them here, they are kind of root, and they're arranged in a circular fashion around the vessel because it's the diameter that matters. The length of it is not important. So you don't want to contract those and decrease the length of the artery. That doesn't help us at all. Okay. Oops. Oh, shoot. Okay. So that is our smooth muscle, no, our artery. Veins are similar. The big difference between arteries and veins is there's a lot less connective tissue. They're a lot thinner walled, but they also have smooth muscle and endothelial cells on the inside. And another difference with veins, as you probably know if you've had anatomy, is they've got one-way valves because the pressure is really low on the venous side. So the one-way valves make sure that the blood keeps flowing back towards the heart. And again, we'll talk about this more later in this class. Okay, and here is a cross-sectional look at our arteriole spindle-shaped cells aligned around it. Here is elastic tissue, and then your endothelial cells, and more connective tissue out here. Okay, so when the cell actually contracts, it's due to a cytoskeleton, right? We talked about this in cell biology if you had me, that whenever a cell moves or something, it's the cytoskeletal interactions that do it. So if you look at smooth muscle cells, they've got these dark spots, dense bodies. Dense always means like electron dense. So when you hit it with an electron microscope, there's dark areas there, and that means protein. So there's these dense areas of protein here that are like little spots here and there. And these are connected to each other by intermediary filaments. So this is kind of like a, well, it's cytoskeleton, but it's kind of near the surface of the cell. And what happens then is when the actin and myosin is ready to contract the cell, the actin attaches to these dense bodies, and then the myosin attaches to something else, and then they pull. And when they do, it scrunches up the whole cell. And so it's kind of like the actin and myosin are pulling on this cytoskeletal structure of the dense bodies with the intermediary filaments attached. And it's important to keep in mind that in smooth muscle, it's smooth, it's not striated, because we don't have sarcomeres. We don't have a regular arrangement of thick and thin filaments that you can see in a microscope. The thin filaments and the thick filaments are when the tissue is relaxed, are disorganized totally, and then, well, not totally, but pretty disorganized. And then when when it goes into contraction, or as it's going into contraction, it organizes, and then it latches on, the myosin latch onto each other, and then they grab the actin and start to pull. So we'll talk about that. Keep an eye out for that. Your myosin molecules, let's see what my figures are. Oh wait, before I get into myosin, here's a picture of the innervation of the smooth muscle. So like I said, these sympathetic neurons are going through that adventitia and surrounding the smooth muscle. And, we always think of axon bulbs as being at the end of an axon. But here you can see all along this axon, there are varicosities. And each varicosity then is like an axon bulb, and it's got, vesicles full of norepinephrine. And so when an action potential comes down here, you get norepinephrine released all over that smooth muscle for contraction. IP3 calcium. Okay, so here is a cheapo PowerPoint animation I made to talk about contraction of the smooth muscle cell. But before I do this and actually see the mechanism visually, I want to talk about it. And all the proteins that are important to it. If you're on Moodle now, it might be worth pulling up this Lecture 7 Smooth Muscle file so we can go through it together, because it might get confusing if I'm just shouting words out at you here. Let's see. Okay, so cytoskeletal proteins involved in contraction. Number one, myosin. Myosin molecules are, usually you think of them as like big it's a dimer, right? And you got big globular heads at one side, and then like a coiled coil between the two molecules of the dimer. So a single molecule would be a long tail, and then a hinge region and a globular head. And this is like two of those with them wrapped around each other. So you got two globular heads. But in smooth muscle, it's like that, but the tail kind of folds up on itself. So it's like, not attached to other myosin dimers. It's just all folded up when it's in the quiet form. So in the relaxed state, it's unfolded and disorganized until you control the myosin light chains. So again, I think I don't know if we talked about this here, but those globular myosin heads have smaller proteins attached to them called the myosin light chains. And the myosin light chains are really vital in terms of controlling the ATPase activity of the heads. And here in smooth muscle, they're also going to control the ultrastructure of the protein, because the myosin will be all curled up until those myosin light chains get phosphorylated. Then it'll lay out, and the tails of different myosins will attach to each other, and then you get these thick filaments. So myosin light chains are key to smooth muscle. Phosphorylating and dephosphorylating is correlated to contraction and relaxation. And like skeletal and cardiac, the light chains are less important. I mean, I believe phosphorylation of them gives you a stronger contraction, but not nearly as important in smooth muscle, where it's like contract or relax based on the phosphorylation site of those myosin light chains. Okay, actin microfilaments are connected to the cytoskeleton, and they attach at the dense bodies at the surface. I think it's really cool how sometimes I feel bad because I feel like I talk about the same things in every class, but everything's connected. So here, these things are connected to these, what are they called? Oh, I can't even remember now. They're junctions that we talked about in developmental biology last spring. Adherens junctions, that's what they are. So they're connected to these adherens junctions, which are then connected to intermediary filaments. And also with actin here, there's tropomyosin on it, just like there was tropomyosin in cardiac muscle as well. In cardiac muscle, the tropomyosin was covering the myosin binding sites, and controlling the position of that tropomyosin was controlling the contraction. That's not true here. It's unclear what exactly tropomyosin is doing, but it's not controlling the actin. It is there attached to it, and I think it kind of coordinates the binding of other proteins to the actin that's going to be important for controlling contraction, but it's not covering the myosin binding sites here. Other proteins that are important now, calponin. Calponin is a small protein that's bound to the myosin and is going to inhibit the myosin ATPase. So this would be in the relaxed state, calponin will be attached to the myosin, and that'll stop contraction from happening. So we're going to have to do something about that to contract, right? A little foreshadowing there. Caldesmin, another important. This is doing a similar thing, but it's on the actin. So caldesmin kind of acts the way you expect tropomyosin to act, in that it's blocking the myosin binding sites on actin. So I think tropomyosin probably coordinates that interaction. But what you really need to know, though, is that caldesmin in the relaxed state is blocking the myosin binding site. So we're going to have to move that to contract too. And the next protein, which is more signaling, I think. Yeah, I should probably put that one down in signaling. All right, let's go on with structural. So myosin light chain kinase. Oh, that's kind of signaling too, all three of these. But anyway, so the next protein we're interested in is calmodulin. So we've talked about calmodulin already. When we talked about the IP3 calcium pathway, I said calcium increases and it binds to calmodulin, and then calmodulin does something in the cell. So now we're going to see what does something in the cell means for smooth muscle, because that's what's going to be controlling your contraction. Calcium binding to calmodulin, calmodulin activating other things. What it's mainly going to activate is the myosin light chain kinase. So MLCK, chain kinase, yeah. So calmodulin will bind to myosin light chain kinase and activate it. Myosin light chain kinase then will phosphorylate the myosin light chains and cause that myosin to open up and then the contraction to happen. Another really important protein here that's regulated is myosin light chain phosphatase. And it's important that it's regulated because typically, I always say, there are all kinds of phosphatases in the cell. And if something is phosphorylated, just assume there's a phosphatase there to take that phosphate off, which is kind of true for the most part. With all our cyclic AMP mechanisms, and we never talk about what phosphatase takes the phosphate off because those phosphatases are always there and they're always active. So to keep the mechanism going, you have to keep phosphorylating, keep cyclic AMPI, keep activating protein kinase A, keep phosphorylating. And as soon as you stop that, then the phosphatases take the phosphate off. Here in smooth muscle, that phosphatase is going to be regulated, which is really important to holding tension after you get rid of the calcium. So the muscle can go into this like long-term contractile state without using calcium, without using much ATP because of controlling this myosin light chain phosphatase. So we'll get into that too. It's all foreshadowing, people. Okay, signaling molecules involved. So we've got two different ways we can contract. One is calcium dependent, and that's the main mechanism, and the other is calcium independent. So the calcium dependent is a lot of what we've talked about already. Phospholipase C, IP3 calcium, calmodulin, myosin light chain kinase, phosphorylating myosin. So that's all the calcium dependent. And we'll also control the actin fibers in this calcium dependent mechanism as well. And then we've got calcium independent mechanisms, which involve some new proteins that, frankly, I just learned about. And they are MLCP and CPI17. So these are inhibitors of myosin. No, they're inhibitors of the myosin light chain phosphatase. So when we control these, we're going to control the contraction by stopping the dephosphorylation of myosin. And this is all controlled by a G protein called RhoA, and the kinase that it activates, ROCK, RhoA kinase, ROCK, R-O-C-K. R-O-C-K in the USA. I'm sorry, I shouldn't have done that. That was really bad. See, I'm alone in this room, so it's like, oh well. Phosphorylation, yes, so ROCK kinase will phosphorylate these myosin light chain phosphatase inhibitors and inhibit myosin light chain phosphatase. So we'll talk about that mechanism in a minute, but in general, this calcium independent mechanism involves going through a small monomeric G protein, RhoA, if you remember RAC-Rho, CDC42 from cell biology. So Rho, which if you remember, was involved in stress fibers and a similar function here. So Rho activates Rho kinase, and then that phosphorylates the myosin light chain phosphatase inhibitors. Okay, so those are the players. Now let's talk about how it actually works. You're going to be really blown away by this intense animation I have here. Okay, so up here we've got a hormone interacting with the receptor, and when it does, the receptor then activates the G protein. The G protein then activates phospholipase C, right? We've done this already. Phospholipase C then will break down PIP2 into diacylglycerol and IP3. So if we follow this should be IP3, I don't know what happened there. So if we follow the IP3 side of the pathway, IP3 then activates the IP3 receptor, releasing calcium. Here's caldesmin, here's myosin light chain kinase. Where's my calmodulin? Oh, it says CD, should say CM. This is calmodulin. So calcium comes out, binds to calmodulin, and then it makes this calcium calmodulin complex, which then binds to myosin light chain kinase, activating myosin light chain kinase. So here's our myosins over here. These are, here's your globular head, where the ATPase activity is. These little yellow things are the myosin light chains, and here's the tail. So the tails are all wound up together. So myosin light chain kinase comes along, phosphorylates those light chains, and then they straighten out. Once they straighten out, They hook up with other myosins and they form these kind of thick, kind of like thick filaments, but not as regular as in skeletal and smooth muscle. But now you can see they're attached to each other, so they can grab on this side and pull to create tension, and grab on this side and create tension. It's kind of like if you're in the water, right, and you're floating, you need to grab on two things to be able to move. If you just pull one, you're going one. I don't know. Anyway, bad analogy. But anyway, let's go to the other side because we've got to get rid of our caldesmin. So up here, DAG activates phospholipase C. Nope, no, I'm sorry. This should be another mistake. This should be PKC, protein kinase C. I'll fix this and upload it again. But this is protein kinase C, which then will come over here, and here is caldesmin, which is blocking our thin filaments. So these purple things are actins of the thin filament. So diacylglycerol activates protein kinase C. Protein kinase C then phosphorylates caldesmin. Caldesmin comes off, and now you get actin-myosin interaction and contraction. So we've got two different sides to this mechanism. One is the calcium side, and the other is the protein kinase C side. Boy, that annoys the hell out of me. Let me go back to that. That's a dumb mistake. Okay. On to Marigold, Sacramento. Okay, that's close. Not exact. This is why it takes me so long to do a lecture, because I can't stand it just being close. It has to be right. It has to be exactly right. Watch this. You're learning my tricks here, which makes me a little uncomfortable, but I'll let it go. Okay. Fill light upper. Okay, now just back here. Oop, wait there. Outline, no outline. Come over here. Okay, protein kinase C. I apologize. I will talk to my boss and have some of my salary subtracted this month for having done that. Okay, let's go back to this. Okay, let's do it again. A little faster now. So hormone binds to receptor. Receptor activates G protein. G protein activates phospholipase C. Phospholipase C breaks down PIP2 into IP3 and diacylglycerol. IP3 will then activate the IP3 receptor, causing calcium to be released from the SR. I'm sorry, ER, smooth end plasma reticulum. Calcium will bind to calmodulin, should be KM there, or CM, I mean. Calmodulin will then activate myosin light chain kinase. Myosin light chain kinase phosphorylates the light chains, causing the myosins to stretch out and organize. And on the other side, the diacylglycerol will activate protein kinase C, which phosphorylates caldesmin, releasing the thin filaments, and then you get development of the contractile apparatus. Now it's all set up, and the contractile apparatus pulls on the dense bodies in every direction, and that causes the whole thing to shrink. Okay, let's stop this crazy share. Let me see your faces real quick. Saline Ramos, Mike West. Nobody else is willing to share? Oh, there's Ariana. See, this is what I miss about being in person, because then in person I can always see you and tell whether you're paying attention or not, or whether you understand or not. Do you guys understand this? Was this okay, or should I do it again? This is good? Good? Okay, good. All right then. So let's let us move on. Oh, there's Matt. Go, Matt. Go. What are you folding laundry, Matt? Looks like you're
About to
folding.
make some food.
You're making
food.
Cool. What are you having for breakfast?
I'm gonna make some eggs.
Nice. I think I'm going to do that after we close up here. Okay. Let me take a quick look at my notes and see where we're headed from here. Okay, so that's one way of causing contraction of the cell. Let me just share the notes real quick because I think I don't have a video a PowerPoint of this. Another way of causing contraction. Okay, so ligand initiated is that whole thing, G protein coupled receptor, and blah, blah, blah. Okay. Another way is stretch activation. So if you think about what an artery does, it's always being stretched and relaxed. And if your blood pressure goes up, even if you're away from the pulse pressure area of the arteries, even in the arterioles where pressure should be constant, it can go up, So say your brain is especially important for this. Say you're sitting down normally, and then you get up and start walking around. Your blood pressure goes up. It automatically goes up to get more blood flowing. So you get that higher pressure going to the vessels of your brain, and they stretch. Well, that's not good. You're getting too much blood to your brain, you're going to get a headache. That's what causes headaches. So as your vessel stretches, it contracts against that strain to keep blood flow constant. That's called myogenic tone. It's myogenic, the contraction is coming from the muscle itself, not from the sympathetic nervous system. So how does that happen? I actually did some work on this when I was I did a postdoc at UVM and I was working on this. So what happens is when the vessel stretches, it stretches the smooth muscle cells, and that stretching causes the opening of some calcium channels, not the Wolff-Spinner calcium channels. These are stretch activated. Some of them are called TRP channels, TRP. But anyway, these channels open and they let a little bit of calcium in. And when they do, that depolarizes the membrane because you've got a positive charge coming in. That calcium coming in then depolarizes, and that opens your voltage-dependent calcium channels. Now, these are really important because we've got a whole class of drugs that block these to cause smooth muscle relaxation. So these VDCCs then are really important for controlling the calcium inside the cell and therefore contraction. So just to go over the mechanism again, the cell stretches, a little bit of calcium comes in through these leak channels that activates the voltage-dependent channels, more calcium comes in, and then you're back into the calcium calmodulin system and the cell contracts. Okay, so pretty simple mechanism, but that's pretty important too. And like I say, we use a lot of calcium channel blockers to control blood pressure. So that is important for us. Okay. Now. Okay. Now the calcium independent mechanism. I said it has to do with the small monomeric G protein Rho. So here's Mr. Rho over here, or Miss Rho, let's not be sexist. Miss Rho over here. Hormone activates receptors. So this is the crazy thing for me. I don't know if you've had me for cell biology. I'm gonna rock your world right now. This is a G protein coupled receptor that's activating a small monomeric G protein. So if you remember back to cell biology, G protein coupled receptors always activated the large heterotrimeric G proteins, the ones with the alpha, beta, gamma. Here's an example of a G protein activating a small monomeric G protein, RAC. So like back in cell biology, we talked about RAC-Rho, CDC42. We also talked about RAS a whole lot. Those are your small monomeric G proteins. So this is kind of like everything, every time you think you know something in biology, you learn more and you find out, yeah, I don't know anything, really. It's all kind of new. Okay, so our G protein then is activated by a muscarinic receptor, actually. And that then activates the kinase ROCK. And, oh wait, let me back up and start at the start. Let's start at the start. Okay, so over here, this is during contraction. Your myosin light chain phosphatase is taking the phosphate off these myosin heads, myosin light chains, and that causes them to relax and let go and fold back up. So this is relaxation going in this direction. myosin light chain phosphatase, taking the phosphate off, myosin letting go. So we're going to activate contraction without calcium now. This is your calcium free. Activate Rho. Rho activates ROCK, which is just Rho kinase. Rho kinase then activates MYPT1 and CPI17, and these block myosin light chain phosphatase. So if you block the phosphatase, you no longer dephosphorylate the myosin light chains, and so you maintain contraction. So this is one of the ways that smooth muscle can maintain contraction even in the absence of calcium. This is a really cool figure. I found this on the web. This kind of ties up, shows you actually how that how the contractile apparatus actually causes the cell to contract. So over here, it's like everything we just talked about, just a different way of looking at it, and more detail, which you're not responsible for. So over here is our G protein coupled receptor, acetylcholine activated muscarinic receptor that activates Rho A. Rho A then activates ROCK. Wait a minute. I should have looked at this before I showed it to you. All right, let's not worry about Rho. Let's just talk about it in terms of the myosins here. So here's myosin. These orange things are the light chains. They're phosphorylated. So the myosin stretches out. Other myosins interact tail to tail with it. And then here's your actin. Myosin starts to grab onto the actin. And then you've got myosin, big myosin molecules here, grabbing actin here and grabbing actin there and pulling. And that's shrinking the cell. So you got your cross-bridge cycling, shrinking the cell. I think what I like about this is this part of the structure here. You can see these are the dense bodies here. Okay, so you've got this actin microfilament attached to the dense body, and here are the newly organized myosin molecules here, and they're grabbing actin attached to different dense bodies. So you're pulling the dense bodies closer to each other, and since they're all interconnected, they all come close together and the cell gets smaller. Adhesosome. Cool. Oops, didn't mean to do that. Okay. Okay, people, that's a 45-minute lecture. Took me about 15 hours to put together, believe it or not, amongst all my distractions here in Pennsylvania. Any questions or comments? Can I see your faces again before we say goodbye? Matt Gamelin. Can we see Matt Gamelin? See, I think he just logs on and walks away. That way we think he's on, but he's not. Doesn't matter to me. Okay, people, that's all I have for today. We will meet back here on Thursday. Everybody feel like they're up and on it at this point? I will hopefully grade your exams today. If not today, then tomorrow. And I will have them for you by Thursday. Okay? Questions, comments, nothing? I'm going to let you go. Somebody has a question, we're going. Peace out, Matt Bernhardt. See y'all later.